Hello, and welcome
Okay.
to the Illuminating Immunity Virtual Summit, Light and Photobiomodulation versus COVID-19. I'm Dr. Joe Doduro, your host, and my guest is Dr. Liisa Laakso. She's the Executive Committee member of the Australian Medical Laser Association and past president of the World Association of Laser Therapy. She's an honorary associate professor and senior research fellow allied health at the Mater Medical Research Institute in Brisbane, Australia. She's a professor of physiotherapy at the School of Allied Health Sciences at Griffith University at the east coast of Australia. She was the first person in Australia to complete a PhD investigating photobiomodulation, and that was some 30 years ago. Really brings a wealth of knowledge to this topic. Thank you for your participation, Liisa.
Thanks, Joe. It's great to be here, and I'm looking forward to our chat.
Well, I am too. I mean, it's been very much a fevered, activity level at the research level. And I knew, talking to the brilliant scientists and researchers when we were doing the Brain Summit, that there was just overflow of information of them wanting to share about photobiomodulation and illuminating immunity. How did you feel that turmoil? How did you get sucked into like this whole thing? What's your thoughts on that?
Yeah, look, it's my interest in photobiomodulation and immunity, I suppose, goes back to inflammation as such, because the two are obviously interlinked. I've done some work around inflammation, chronic inflammation and acute inflammation in animals, models, benchtop animal models, trying to understand what the mechanism is for photobiomodulation in acute pain settings and chronic pain, and looking at the release of endogenous opioids at the local and regional level, as opposed to the systemic level, which was the focus of my research when I was doing my PhD. I looked at beta endorphin and ACTH plasma levels in people with chronic pain at that time. And at that time, 30 years ago or so, we didn't really have all of the kinds of technologies that we have now for investigating those things. So I hypothesised at that time that there was some link through the stress immune system between the peripheral application of PBM in people with chronic pain and the central nervous system through the hypothalamic pituitary adrenal axis. And it's interesting now that we're starting to see those kinds of things being verified in literature. So it's not a big leap for me to be thinking about inflammation or infection, endogenous opioids, cytokines, those kinds of things. And as my background is in physiotherapy, I'm really very interested in the pulmonary effects of inflammation. So that's kind of it in a nutshell, Joe.
For me, when I'm starting to study the mechanisms, because that's kind of what we do, what's the mechanism of this disease? And when you start to hear, cytokines and interleukin this and, T cell this, and you're like, hey man, I'm reading that in my literature every day. What I'm doing has an effect on these. And I think that's sort of what has, gotten these scientists up and raise their hand and say, hey, this is something there's a connection here. Am I correct?
Yeah, look, and there has to be when you think about it, when you go back over the history, and I think that's been the interesting thing for me in reviewing this paper that I'm going to be presenting today and discussing with you, are those historical links with what is happening these days. And the body is a fantastic machine, if you like, and so much of it interacts and integrates, and there are all kinds of things that come together, and they can't be teased apart. They do work in conjunction with each other, and why shouldn't something like light have an effect on some of those systems that we know from the past have been influenced by light.
Remember, we were talking in that we're actually attempting to treat the human, not the disease.
Yeah.
Which is, like I said, sometimes it's hard to get people to understand it, but the mechanisms are great. Shall we go to the slides?
Sure thing.
So we're going to be reviewing a recent publication from Enwemeko Chichuku, his lab at University of California, San Diego, I believe. Light as a potential treatment for pandemic coronavirus infections, a perspective. Take it away.
Thanks, Joe. Yeah. Really what I'm going to try and do through this next little while is look at the paper in some detail and then provide some reflections at the end of that, and perhaps propose an idea out of those reflections that might be interesting to researchers in general. Now, I have to disclose that with a physiotherapy background, I don't have a benchtop science background. I don't pretend to know all the ins and outs of all these things, but I've worked with some great people in the past, and some of my research has been at that benchtop and animal level as well as the clinical level. So I'm hoping that I can bring something to this discussion through that experience. So this is the paper, a snapshot or a screenshot of the paper itself, and you can see there the title. It's in the Journal of Photochemistry and Photobiology B, and the author group there. If we think about the paper itself, it's not a randomized controlled trial, it's not a systematic review, it's a perspective, and it's primarily based on blue light. And so most of the conversation or most of the perspective within the paper was on blue light. But having said that, a lot of the detail and a lot of the research evidence that is presented in the paper is not necessarily on blue light, and you'll see that as we work through here. The authors themselves are very well known in the photobiomodulation field, particularly in wound healing, and Chikuka, Professor Enwemeko, has, according to ResearchGate, recently 111 publications. I thought that there might be more than that, and because I didn't have access to some of the other ways that you measure research outputs, that's the best I can do. This group has been collaborating for several years if you look back at the history of their papers together. So let's go to that paper. And the first part of that paper is devoted to the history of light and pandemics. And when I was looking for some illustrations in that area, it was interesting to see from the Spanish flu era 100 years ago. This is a sports group wearing influenza gauze masks, and we know about the issues around masks these days. Are they protecting people from someone else's aerosolized viruses, or are they protecting you if you're wearing them? So there's a bit of back and forth around that. But, a lot of the things that we're seeing at the moment around public gatherings, the numbers of people in public gatherings and all those kinds of things are really a throwback to what was seen a century ago. So the Enwemeko paper starts with that historical perspective, looking at pandemics of the last 100 years. And they make the point that along with face masks and social distancing, that phototherapy was one of the most effective tools used to minimise the impact of the 1918 pandemic and in other disease pandemics or epidemics, and it's been rather overlooked in our most recent pandemic. In the 1918 flu pandemic, from the Enwemeko paper, it was pretty clear that at those times, in less sophisticated times perhaps, that sunlight was effective in reducing flu-related mortality and morbidity and person-to-person infection, and that people recovered better, and that it prevented the death among patients and infections among healthcare workers at that time. So they were interesting reflections to start with. The authors of the paper then go on to talk about heliotherapy in Egyptian and Greek and Roman times, and they go on to talk about the development of artificial light sources in the late 1800s and early 1900s that were used probably throughout the 1900s, but once antibiotics were being developed, then those light sources kind of fell out of favour. Those of you who know this field will recognise this kind of photo, which was Finson's light lamp. Niels Finsen was a Danish physician who won the Nobel Prize in 1903 for the development of that light source to treat skin tuberculosis. It was considered a bactericidal intervention. It was erroneously thought to be emitting ultraviolet wavelengths. And Wemmecker in that paper draws out the error in that and suggests or states that Finson's lamp couldn't have emitted ultraviolet light, but it was probably violet or blue light because the type of glass used in the lamp, and that the tuberculosis bacterium is known to absorb blue light and not so much ultraviolet absorption. So they were interesting background comments. And if we look at some of the further conversation that the authors of that paper go on to make statements, they state that the bactericidal effect of sunlight is often ascribed to ultraviolet. But, you know, that can be attributed to the immense amount of blue light that reaches the Earth from the sun. And you can see there on the left, I've sourced a graph that illustrates that blue light tends to have a greater spectral irradiance. Someone's at my door. I'm sorry about the ringing, but I'm sure they'll go away soon. But you can see that peak transmission at the surface of the Earth is actually in the blue region rather than in the ultraviolet region, and that is 10 times more in the blue region than in the ultraviolet region. So the authors then go on to talk about the mechanisms of blue light on bacteria, and so they propose 4 mechanisms, the first of those being the most likely, that blue light triggers endogenous bacterial chromophores, particularly using wavelengths between 405 and 470 nanometers. That results in ROS, oxygen species, and that if enough of these ROS are activated, then that will result in cell death. And a lot of that work has been done by this group of authors themselves. So I'm going to look at that last paper there, paper C, which is the one about optimising the bactericidal effect of pulse blue light on an acne species. And if we look at that particular, or some of the screen grabs that I've taken from that particular publication, you can see there along the bottom the images that the authors provided in that paper, the control petri dish or agar dish, and then 3 different fluences of light, 5, 10 and 20 joules per square centimetre, and the statistical significance in the reduction of the bacteria with the higher exposures there. So, you know, really nice evidence around the effect of blue light in that particular study. The second mechanism that they go on to talk about was about the fact that blue light alters bacterial cell membrane integrity and alters cellular functions as well. And this time they presented evidence from a paper that they had done with other authors using light of 450 nanometers, looking at the structural architecture of methicillin-resistant Staph aureus. And here again, in a similar way, I'm presenting some of the data from their paper. On the left is the control with the MRSA, and then with a single application of the light in panel B, and a double application of the light in panel C. And you can see the statistical differences with a single application and a double application against the control there in colony count. The third mechanism they talk about is an alteration in DNA, and this time they've used a 470 nanometer wavelength. They've repeated the applications 2 and 3 times, and they've done that in combination with vancomycin, a very strong antibiotic, again treating MRSA. And again, in a similar vein, you can see there the controls on the left, and in the 3 panels C, D and E on the right-hand side of the screen, blue light irradiated colonies of MRSA, ultraviolet light irradiated colonies, and the vancomycin treated MRSA are showing very similar outcomes in relation to the ability of light compared to a strong antibiotic medication to reduce methicillin-resistant Staphylococcus aureus. So I think that that evidence is really quite convincing there, at least benchtop-wise. The fourth mechanism that they talk about is the upregulation of prophage genes. And this is from a paper from a different group of authors now who used 460 nanometer blue light as well as 630 red light. And their conclusion was that about 30% of upregulated genes were phage-related or phage-related genes, depending on what school you went to. And if we look at those results, and again, I'm not, as I say, I'm not a scientist, but you can see that there was a significant differential in the genes that were expressed in the MRSA genome after the blue light irradiation, and that was specifically upregulated in 16 of those genes using real-time PCR. And this is another graphical representation of that, where they used blue light with 2 different fluences there, 60 and 120 joules per square centimetre, comparing that against the controls. So there seems to be something there in terms of the genetics of the effect of blue light in these bacteria that have been tested thus far. So I'm just going to pause there for a moment. Joe, is there anything that you wanted to raise out of any of those initial slides?
Well, I'd have to say that that's exactly what I was looking for, because I've seen Chichuku's group present this research at conferences, and I'm totally blown away by the effectiveness for the bacterial load that they looked at and the MRSA, which was at that time when he presented that, MRSA was unkillable. And there he is presenting the research that says, you know, I put the blue light and we killed 98% of the MRSA, boom. And I was like, wow, that would save a lot of lives if you just treated all the stuff, you know, the hospital setting, you know, not the UV, the blue light, the ones that he said. And then maybe we'll come back to that. But I think that that outlining those mechanisms was very, very key, because, you know, we're looking at it on a cellular, you know, in the bench type of things. And we want that to reflect also what we're seeing, you know, theoretically in a clinical setting. So I appreciate that. And I'm going to touch on the phage upregulation. That's like the ability to get rid of the macrophage type of thing, correct? Would I be correct?
Yeah. Yeah, well, it's the killing ability, I guess, of those cells. So, yeah.
Good.
Okay, so ready to go again? So moving on to understanding what the authors were thinking in presenting these original data around blue light and bacteria, they transitioned on to thinking about whether there was any effectiveness for viral loads as opposed to bacterial loads. And they posited that the use of light might be highly beneficial in managing opportunistic bacterial infections in people with suffering from viral infections, giving the immune system a better chance of overcoming deadly disease. And I think that that's something that theoretically a lot of people are in agreement about. So let's have a look and see, regulate, if you like, or blow that up a little bit more into clinical settings.
One question that I think that seems to be pertinent on that is that it's true. You know, there's bad bugs in the hospital, and they're not, I don't believe they're viral. These are bacterial things that give you the superbugs. They're bugs, they're bacteria. So I definitely had this thought that, you know, one of the worst places for sick people to be is in the hospital. And then if they're pulmonary, you know, deficient, wow, right? With all the super cleaning that they're doing, sometimes that makes the super bugs. So I think that he's touched on that, and I think his research in the MRSA probably lended itself towards this also. But I'm interested in your take. Please continue.
Yeah, look, I think it's really important for us to be, again, thinking about the whole human. We're not just targeting one virus here. We're targeting a whole human who may be, depending on the severity of the disease, may be on the escalator towards severe coronavirus infection results. But maybe if we can prime the immune system better, or treat those opportunistic bacterial infections, which in themselves create the inflammatory response and the immune response, then perhaps that's going to reduce the effect of the extra viral infection as well. So I think there's lots of things there for us to be thinking about, and that really focusing too much down on one aspect of this particular pandemic is probably, it's probably natural for scientists to do that, but it may not necessarily be the exact right thing that we should be doing from a broader clinical perspective. I'll move on and we'll perhaps look at light and pulmonary disease. The authors do start to talk about this, but in setting the scene here, I just wanted to reiterate what a lot of people already know, is that the lungs are the primary target organ of this novel coronavirus, the SARS-CoV-2 virus, which is causing the COVID-19 disease. And a cytokine storm is really what's putting people into intensive care settings in those severest of presentations. So a really uploaded inflammatory reaction within the lungs. So, and Wemaker and co-authors go on to talk about the fact that the existing antivirals are ineffective in this particular COVID-19 disease. But there is some information in the literature that suggests that light photobiomodulation may be something to consider in pulmonary diseases. And they cite a number of studies that point the way towards this. So in this particular study, in rats, they used red light. The authors are listed there at the bottom there. And 1 hour after induction of the pulmonary inflammation and the treatment, they found that it inhibited pulmonary edema, downregulated neutrophils, and reduced the damage to cytoskeletal elements, and reduced TNF, tumour necrosis factor alpha, and reduced interleukin-1 beta, which are a couple of the cytokines that you would expect to see in an inflammatory response. Now, I've highlighted TNF alpha there because I want you to remember that when we come back a little bit later to talk about some reflections that I have from this paper. So that's red light. In that particular study, Along the bottom there, this was lipopolysaccharide-induced pulmonary inflammation. You've got the control image there. You've got the LPS, which is the lipopolysaccharide circumstance, which is the big black bar in the middle there, and the neutrophil count. And then on the two right-hand side bars there on the graph, you've got the induced pulmonary inflammation with the PBM, the photobiomodulation treatment, and the same induced pulmonary inflammation treated with a corticosteroid. And you can see there that the effect of the light was just as effective, perhaps even better than the corticosteroid. And corticosteroids in this COVID disease have been considered to be something that are inadvisable for a number of reasons, and I won't go into those, but my clinical colleagues in intensive care tell me that the application of corticosteroids in patients in intensive care with COVID-19 is not advisable. So here's an opportunity to be thinking about something else instead of the routine or standard of care that would normally be applied, which is corticosteroid. Another paper that the Enwemeka authors talked about was a similar kind of design, in this time inducing pulmonary fibrosis in mice, treating the mice with red light again, and noting that there was a reduction in collagen, in alveolar inflammatory cells, in interstitial thickening, and there was actually an increase in pulmonary elasticity, a decrease in pulmonary elasticity, which to me was counterintuitive to what I would have expected. But let's look at those authors' results again, presented in a similar way, and I've just chosen small snapshots of information from all of these source papers. It's too detailed to go into in any great detail in this presentation, but again you can see from histomorphometric analyses in this particular paper, A along the bottom was what the authors call basal, but probably the control group, the photobiomodulation therapy group in panel B, and the second bar in this graph, the pulmonary fibrosis group, which is F, the big black bar in this particular graph, and in panel C, and the fibrosis treated, pulmonary fibrosis treated group with photobiomodulation therapy in the final bar there. And you can see that there is a significant difference between the control fibrosis, pulmonary fibrosis group of animals compared to the group that is treated, that has had induced fibrosis and treated with photobiomodulation there. So again, fairly compelling results on that paper. And similarly,
Lisa, if I could, I was very much hoping that someone would address this topic, because let's say you were infected, you almost died, but you come back, you recovered, everybody's cheering, right? But you have lung damage. You have fibrotic lung damage. You may have a dry cough. You may have a respiratory illness for the rest of your life. And I was like, I think we can change that. You know, I think the parameters said something about fibrogen, and we have these things. And here's some research that shows that this may be complementary along while you're having the infection, if you will. But afterwards, maybe we can change these lung fibrosis, these things that are, that's going to be a chronic disease if there's not some approach. Would you agree?
Oh, absolutely. And I think we're starting to see now some guidelines around the rehabilitation of post-viral infection in these patients who've had COVID-19. But the other thing I think, which is also as important, is that immediate post-viral period when people may still, that they've recovered from their COVID-19, but they've still got that dry cough, and they may still be shedding some virus. And that's something that's not clear at this stage. in terms of how much virus is being shed by people who are recovered, and whether they are in fact still susceptible to further infection with the virus. So there's still a lot to know about that side of things.
Well, we're catching it. We're doing our, if you want to be a first responder, this is the type of research this is the explanation of what we're going to, possible ways to address this. I really appreciate it. Go ahead.
So moving on, there was one more paper done in animals, this time similar paper, similar design, induced pulmonary fibrosis in mice, this time using an infrared or a wavelength that was kind of on the cusp between visible red and infrared, 780 nanometers. And you can see there the results. I've got them bolded in that section. Similar results there in relation to the effect on cells, the effect on inflammation. And if we look at in a bit more detail at that source paper, you can see that on the left in the bottom panel is the control image. The middle panel, or the large black bar there in the graph, is the bleomycin-induced pulmonary fibrosis. And then with the addition of light or photobiomodulation, there's a significant reduction in the airway collagen, very similar to if you compare the image on the right here in the bottom with the control image on the left along the bottom there. That's looking pretty good from the perspective of airway collagen. And a lot of the other data in all of these papers essentially lines with those kinds of results that I'm presenting here today. So let's think about clinical studies. Where does this put us? We've looked at some of the benchtop work. We've tried to bring this forward into pulmonary settings. There's not a lot around, and I think the authors of the paper that I'm reviewing recognise that in terms of the papers that they've presented in this particular publication. They present a couple of pieces of evidence from source journals that I was not able to access in the English language. I was able to access the abstracts, but there were no specific details in those particular papers beyond a summary of what the outcomes were. So there was no details on the type of light parameters that were used. But these were in human or clinical settings. This first one in COPD and the next one in bronchial asthma. And from the abstracts alone, there is a suggestion that there may be some benefit for using light in human settings.
If I could speak to the first paper, one of the things that I think that we may be looking at, because it was brought up by one of the presenters, about the thrombosis, about the rheology, platelet aggregation, just thinking having more debris in the blood changes its viscosity. And this may be one of the reasons that, the autopsies are finding a lot of thrombosis, and all these organs can be susceptible to that also. So, having a change in the blood via the light, therapeutic light, the photobiomodulation, that might be part of what they're recognizing in this sooner than later.
Yeah, and I think that's been quite a surprise to people working in this field medically, that this is a resultant of this particular virus, coronavirus. And there's a few other really weird things that are happening as well with this coronavirus, which are yet to come to light in terms of consistent findings. And one of those being the potential that this virus is unmasking in children, the Kawasaki disease. So I think there's a lot of water to flow under this bridge, and we'll know much more in the coming months and years. So if we go on to looking at some of the other papers that the authors in the Enwanika paper looked at to try and make the case for photobiomodulation treatment of viruses, they cited some studies that were looking at sample storage of viruses, which I thought were interesting. This particular study was looking at the long-term stability of baculoviruses stored under various conditions, and they found that there was that exposure to light was a major factor in reducing the infectivity of the baculovirus over several months. And in another paper, which was doing a similar thing, I'm trying to understand the stability of a leukemia virus in storage, found that prolonged storage of that particular virus in ambient light led to a loss of infectivity of that particular virus. So kind of a backwards kind of way of looking at the potential for light in reducing infectivity of viral loads. And then Enwanika and colleagues then went on to provide a little bit more evidence around citing three papers that were looking at the effectiveness of photodynamic therapy. But I think I've produced enough in this review of these papers to date to suggest that the authors were making a fairly good case around the potential for light in viruses. They finished off with this final reference, which was a 2020 paper that looked at simulated sunlight, or as the authors suggested when I went to look back at the source paper, broad spectrum light, being tested on aerosolized influenza virus. And they found that there was a significant inactivation of the flu virus. And that was demonstrated through viral decay constants and the half-life of the viruses. The decay of the virus increased with increasing exposure to light and higher intensities of lights, which is really important for us to think about in relation to PBM, that there are probably certain wavelengths, certain parameters of PBM that we need to be thinking about and understanding better so that we can put that into clinical models. The other thing when I went back to that source paper was that the authors of that paper felt that there were a couple of limitations in their work, and one of those was the effects of the culture medium that they used on the flu virus that they were using, and the fact that they were not using wild-type viruses. There may be different effects, or perhaps they could be the same effects, but they don't know what the effects might be on wild-type influenza viruses or other viruses. So I think there's some important work there to look at in the future. So the conclusion by these authors was that there was urgency to explore the potential of blue light specifically as an antiviral agent, that it could be used against opportunistic bacterial infection with people who are infected with viruses, and that it could also be used to disinfect equipment and environment and spaces as well. And I think we've kind of touched on that in our conversation today, Joe. Mm-hmm. No one wants to go to hospital and pick up another disease, and I think that's what people are worried about at the moment, particularly people with chronic diseases. They don't want to go into a clinic or a hospital and then find themselves being put at risk of catching something else, such as this coronavirus. I know that the authors are now advancing their work, advancing the applications of PBM through investigating the virucidal effects of blue light in some viral models. So it will be really interesting to see what their conclusions are coming out of that work that they're pursuing in the next little while.
If I could,
I want to talk a little bit.
If I could, yeah. One thing there. One is, it's kind of surprising that they never really looked at the viral component, until this point, because it sort of lended itself, to me, was always the question, well, it did the MRSA, but anything else? So it's just a systematic way of going through that. And when you think about this, disinfect equipment, environments, and spaces. Wouldn't it be nice if we could go to the bar and know that they killed everything that could be, instead of just hand wipes, but really blasted it? I mean, I think that would be a good thing for everybody. Nobody wants to leave their house because they're afraid they're going to get something, much less go to the hospital where it's full of the superbugs. So I'm with you.
Yeah. Look, I think I don't blame the authors for concentrating on MRSA. It has been a really problematic issue particularly in clinical and hospital settings. And their work has been focused really nicely on that bacterium as well as others. So I think, from a science point of view, I understand why they've been concentrating on bacteria. But they're now, and this is what the pandemic is allowing us to do, is it's allowing lots of researchers, lots of scientists to pivot and use their knowledge from other areas and apply it, fitting it for purpose in this current pandemic. But I hear what you're saying, Joe. I think, these are the kinds of things that we can learn out of this. If I then go on to just one slide on my own reflections from this perspective paper, and we have to remember that this is a perspective paper, that not necessarily all the literature in this space has been covered, but I think I learned a lot from going back to the source papers, and some of that I've tried to convey to you today. And I think some of the other speakers in this virtual conference will be speaking to some of that other evidence. From my perspective, the paper brought together really nicely the case for testing blue light against viruses, and these authors are going on to do that. I think that it also demonstrates to me that we still have a long way to go before the case can be made for funding a rigorously controlled and adequately powered clinical trial. I think there's still some things that we have to do. And some of those things are around identifying relevant biomarkers, and that's best done, I think, at that benchtop stage. It's best done, I think, for identifying the parameters for use in vivo studies, and in vivo studies we can better test those parameters that have been identified in benchtop studies, and then identify the physiological markers that might be transferable into clinical studies. And our early clinical trials going forward, we need to be testing the right parameters and the right clinical markers, because clinical studies, as we know, are really expensive, and there are lots of ethical issues that sit around that. And I think in the middle of a pandemic, we can't forget what those ethical issues are, despite the urgency of finding treatments. So I wanted to present an idea here from reading this paper and reading the source papers that underpinned these authors' thoughts. And I wanted perhaps to suggest that we use TNF-alpha as the clinical biomarker. I understand that there may be people globally who are now looking at using photobiomodulation, and Joe, you probably have something to add to that. But perhaps TNF-alpha is the primary biomarker. And the reason I say that is that there's some evidence from outside of our field in zoonotics that demonstrates that bats use the antiviral cytokine interferon alpha to combat viruses. They are well provided within their physiological systems with this particular cytokine. And perhaps it is the key to preventing the bats from being affected by coronaviruses. If we look at the evidence in photobiomodulation, it shows that red light specifically can modulate TNF-alpha. And that's been shown in one study that saw an increase in TNF-alpha. If you remember back to the earlier slide, that was shown to decrease, I think it was, TNF-alpha, and others have shown this in unrelated neuroscience research, that there's a reduction in TNF-alpha using red light. So I think we can be using information from outside sources to be influencing or guiding us on where we go with the application of light in clinical settings. And I would suggest that perhaps we think about TNF-alpha because it is so closely related to interferon alpha, which is the bat's shield against coronaviruses. So perhaps that's something that we can be thinking about in relation to these cytokine storms and the inflammatory effects of these new viruses. So lastly, I just want to say we should never stop dreaming. We should never stop thinking that there is a role for light, for PBM, in a lot of the things that challenge us in healthcare, but we also need to stay grounded in reality and to do things in a tiered way, in a way that helps to progress the field and allows us to convince the skeptics that PBM is in fact something that can be really useful, and we're not being quacks and cranks and whatever else in suggesting that light alone has an effect in infection and inflammation. So I'll finish there, Joe, and I'm happy to spend a bit more time having a chat or leaving it at that.
One question I had is that, we're looking at the dose just as an aside here. So we've seen several different wavelengths or colors of light having similar effects. I saw 250 milliwatts, 150 milliwatts, and some very low 3.6, joules, 250 joules and 150 joules, and then like 6 joules. And so it's sort of, again, the same scenario that, more light may be better because, if we're having a photobiomodulation effect, meaning all these immune changes and the body started to gear up and the genetic changes and what have you. But maybe if we had the ability to put more power and more light, the photodynamic effect, which is the killing of the cells, whichever, healthy or sick or what have you, maybe there's a little bit more of a window for these things, just based on the different numbers that we're seeing, because no one has really discussed killing the virus or killing the bad cells that, I think that's basically what the blue light was doing, it was just killing those cells at, somewhat a high power for a little bit.
Or preventing them from multiplying, Joe, preventing them from multiplying, which is a different, that's the modulation part, the influence of inhibition as opposed to killing, which is higher up in the Arndt-Schultz curve. So I think there's a lot of discussion to be had there. There's a few things that I could say to that. Those models were primarily in small rodents, so the small amounts of irradiance and fluence that were being used are probably appropriate for those small rodent models. We may need greater amounts, intensities, if you like, in human clinical settings. But I'm not necessarily convinced that we need to go over the top either, because from my early PhD work, I found that if you over-treated, then you very quickly led to an accommodation, if you like, or an inhibitory response on pain. You needed a certain amount, and you needed to do that a few times before it triggered a physiological response. That physiological response occurred before the clinical response occurred. But there's also other research in other parts of our field that suggests that you can over-treat. And I'm not necessarily convinced that high irradiance is the answer, but I'm happy to be convinced in different settings that it's a necessity.
Yep.
And they're the kinds of things that we don't really know yet. If we're causing an increase in cytokines in a clinical model and we've already got someone who's suffering from a cytokine storm, could we be making things worse? And, you know, there's those kinds of questions that occupy my mind.
Well, that's what, I mean, clinicians are always, you know, first do no harm. I mean, that's our oath. And I think that we see that it will, you know, different wavelengths will accelerate the inflammatory process, step on the gas, but then it has a compensatory step on the brake too. So it sort of has a biphasic effect because, you know, if it's normal, if it's accelerating normal physiology or physiological processes, modulating them in that way, I think that's sort of, it has a built-in brake. You follow me? So it's just,
it's that homeostasis, isn't it?
Right. So it's like if we're looking at what stage of the disease, you know, what stage of the recovery process, you know, pre, during, post, acute, you know, those are probably going to have different parameters. And then the other thing is once you start putting light on them, it's going to change them one way or the other anyway. So yeah. I agree that I know that everybody's busy planning their research and examining these things. And I think that your contribution and the contribution of the other speakers is going to be a very, very good resource for people who want to know, you know, what are the mechanisms? How does this work? What's the possibility? Just so that they can, you know, see the light, the illuminating our immunities. You have any final thoughts you want to put out there?
Not really. I think I've covered everything that I wanted to cover. I think the paper was a really well-written perspective paper. I think they've been balanced in trying to bring forward what is known anecdotally from history and what is currently known in a more technological digital age around evidence-based practice. You know, the next step is really if we want to use PBM, we really need to be doing some work between this pandemic and the next one around understanding properly what kind of parameters are appropriate for viral treatment, and maybe that step is using it in influenza situations to help to provide us with some kind of idea around how it could be used in the next coronavirus pandemic, because from all reports there will be one.
So when we are looking at your hypothesis, you're kind of looking as a marker that how the immune system is responding. TNF alpha might give us the little, you know, I'm getting like this, and now I have to treat, and now it can be modulated to do what it's going to do or have an influence on it. I think that's something that people are going to be, I think it's a good way to look at it.
Look, I think if people are going to be working in this area, we need to have some agreement on what it is that we're measuring. And perhaps TNF alpha is something that needs to be a constant in all of those studies. Certainly measure other things, but let's try and use TNF alpha as the key component that is consistent and standardized across all of those studies so that we can do some proper comparisons when it comes down to it.
Oh, that's brilliant. Thank you. Thank you for all your help, your assistance. Great job.
Thanks, Joe. I really enjoyed it.